Hello everyone, welcome back to my new video. So today we will be discussing about the formation of the hemoglobin. In the first part we have discussed about the general property, introduction and the structure of hemoglobin. In this video we will be discussing about the formation of the hemoglobin. So let's get started. Firstly, a little introduction. The synthesis of hemoglobin actually starts in the pro-erythroblastic stage. Now what is this actually? When we have studied the stages and development of RBC, we have seen that there are certain stages. For example, the pro-erythroblastic stage, the intermediate, the early, intermediate and the late normoblast, the reticulocytic stage and all. So while studying this stage, we have seen or we have studied that in the pro-erythroblastic stage itself, the synthesis of the hemoglobin mainly starts. But however, the hemoglobin is not visible in that stage. So the hemoglobin is only visible or appears in the intermediate normoblastic stage. And after the intermediate normoblastic stage comes the late normoblastic stage, where the hemoglobin number increases, and in this way the hemoglobin produces or multiplies and continues to grow in the cell, and it is continued until the stage of reticulocyte. Next, it mainly involves the following stage: the synthesis of heme and formation of globin. Because definitely hemoglobin is made up of two components: the heme part, the iron part, and the pigment, and the proteinaceous part, which is the globin part. So both of them needs to be produced or synthesis accordingly. So we'll be studying first the synthesis of heme. So if we take the example of a diagram, firstly we are taking this. This is the mitochondrion, which is also known as the powerhouse of the cell, and this white part will be called as a cytoplasm. So what is happening that, and worth remembering, that the synthesis of the hemoglobin or the synthesis of the heme first takes place in the or starts in the mitochondrium, or it initiates from the mitochondria. So what is happening that an enzyme, or that is the succinyl CoA, the full form is the succinyl coacetic enzyme. So what is happening is that this succinyl CoA is combining with glycine. So after combining with glycine under the influence of the enzyme, which is known as the ALA synthase, that is the aminolevulinic acid synthase. So succinyl coacetic A or succinyl coenzyme A plus glycine under the influence or the catalytic action of ALA synthase, that is the aminolevulinic synthase, it creates one molecules of alpha aminolevulinic acid. And then what happened is that after the formation of this molecule, this then transfers outside or moves out of the mitochondria into the cytoplasm, where two molecules of alpha aminolevulinic acid will combine together, and under the influence or catalytic action of the ALA or the aminolevulinic acid dehydratase will lead to a product known as the porphobilinogen. And also the porphobilinogen, under the action of another enzyme, that is the synthase, will form the uroporphobilinogen 1. The uroporphobilinogen 1, again under the action of another enzyme, that is the co-synthase, will form uroporphobilinogen 3. The uroporphobilinogen 3, again under the influence of another enzyme, that is the decarboxylase, will obviously the name itself is tells us decarboxylase, because the carbon numbers will be reduced. It will form the coproporphyrinogen 3, and then the coproporphyrinogen 3 will again transfer or move back into the mitochondrium, where this coproporphyrinogen 3 will be oxidized by the help of the oxidase enzyme, and it will form protoporphyrinogen 9. And finally, the protoporphyrinogen, under the help of the same enzyme, that is the oxidase, form the protoporphyrin 9. And lastly but not the least, under the help of the addition of the iron molecule itself, and with the help of the enzyme the ferrochelatase, it will form the heme molecule. And again the heme molecule will again come out from the mitochondrium back into the cytoplasm, where it is combining with the globin part of the molecule, which is already being formed, and finally it will give rise to hemoglobin. So summarizing it up quickly, succinyl CoA plus glycine will form alpha aminolevulinic acid, which will travel outside the cell, and then two molecules of alpha aminolevulinic acid will first form porphobilinogen, then it will form uroporphobilinogen 1, then it will form uroporphobilinogen 3, Next, coproporphyrinogen 3. Next, coproporphyrinogen 3 will travel back into the mitochondrion, where it will be converted into protoporphyrinogen 9, which will be again converted into protoporphyrinide, and finally with the help of an enzyme and addition of iron molecule, it will be forming heme, which will again travel back into the cytoplasm and combine with globin to form hemoglobin. So in this way the heme molecule is formed. Next, we'll be studying about the formation of the globin. So globin is what? Globin is nothing but a proteinaceous structure. So polypeptide chains, polypeptide means there are multiple peptides bond, and polypeptides chain of globins are produced in the ribosome. We all know that ribosomes at the site where the proteins are actually prepared. So there are 4 types of polypeptide chains, namely alpha, beta, gamma and delta. We have definitely studied this while we were studying the hemoglobin. You can definitely check this video. I'll leave a link of the video down in my description below. The adult hemoglobin consists of 2 chains. The fetal hemoglobin is slightly different because it has the different kind of chain. For example, the normal adult hemoglobin consists of the alpha chain and the beta chain, whereas the fetal one may consist of the gamma chains as well. Next, each of this chain differs from others by the amino acid sequence. Definitely, each globin molecule is formed by the combination of 2 pairs of chain, and each chain is made of 141 to 146 amino acids. That means that each of the globin molecule will be forming by 2 pairs of chain, which will be having 141 in one chain and 146 amino acid in another chain. An adult hemoglobin contains 2 alpha chain and 2 beta chain, whereas fetal hemoglobin consists of 2 alpha chain and 2 gamma chains. That means if we consider the an adult hemoglobin, so it is made up of 2 alpha chain and 2 beta chains. Now what is happening is that each of them consisting of 141 and another 146 amino acids. So this is what it is been explained. Next, the structure of hemoglobin. So firstly, as you know, the hemoglobin mainly consists of 3 parts: the iron part and the heme part and the globin part. The heme consists of the pigment containing part, which has an iron part, and the porphyrin, that is the pigment part. So the iron part is present in ferrous Fe2 plus form. It is unstable or loose form. Again, it has to be in unstable form, because if it is somewhat stable, then the body's normal physiological condition will be disrupted and it won't be taking part in any of the physiological processes. Next, porphyrin, which is the pigment part of the heme, it is formed by the 4 pyrrole rings, and since it is made of 4, so the name tetra, thus tetrapyrrole, and they are called as tetrapyrrole 1, tetrapyrrole 2, tetrapyrrole 3 and tetrapyrrole 4 respectively, and they are attached to one another by methane, that is the CH4 bridges. And lastly, the globin, which we have just studied about the synthesis of the globin. The globin contains 4 polypeptide chains. Among the 4 polypeptide chains, 2 are alpha chains and 2 are beta chains. This is in case of the adult hemoglobin. In case of fetal hemoglobin, it will be 2 alpha chains and the 2 gamma chains, so respectively. Next, for configuration, each polypeptide chain combines with one heme molecule. Thus, after a complete configuration, hemoglobin molecule contains 4 polypeptide chains and 4 hemoglobin molecules. Yes, because while studying, we have just considered the adult hemoglobin that consists of 2 alpha chains and 2 beta chains. So 1, 2, 3, 4. Total there are 4 polypeptide chains, and each of them will be binding to one heme molecule. Each of them will have their own individual heme molecule. So what is happening is that there is a total of 4 polypeptide chains and total of 4 heme molecules. So the fate of hemoglobin, that means what will happen. So before explaining the fate of hemoglobin, a final summarization is that an entire hemoglobin molecule consists of mainly 3 parts: the heme part and the globin part. The heme part mainly consists of the iron and the pigment containing part, which is also known as the porphyrin, and there are actually 4 heme molecules and 4 polypeptide chains. Next, the fate of hemoglobin means what happens to the hemoglobin after its life cycle gets over, or what is the ultimate fate of the hemoglobin molecule. So we all know that the lifespan of RBC is 120 days, and after that it gets destroyed in the reticuloendothelial system, which is particularly seen in the spleen, and then the hemoglobin is released into the plasma. So soon the hemoglobin is degraded in the reticuloendothelial cell and split into globin and heme. Now definitely what is happening is that in the reticuloendothelial cells, the hemoglobin is getting degraded into a heme part and a globin part in the reticuloendothelial cells. And how is it getting degraded? This is an important this theory is very interesting. Now why? I have already explained this in my RBC video. What is happening is that in the reticuloendothelial system, actually what happens, the RBC, when it becomes old, or when it ages, it actually gets more fragile, and the spaces between the capillary are very minute, as a result of which the RBC, when it tries to squeeze, because it has a squeezing action, RBC, when it ages, it loses its squeezing capability, as a result of which when it tries to pass through the capillary system in the endothelial cell, it actually gets broken down. So in this way the RBC gets broken down, and what happens is that similarly the hemoglobin gets broken down into heme and globin part, and then the heme is again degraded into, this heme part is degraded into iron part and the porphyrin part. We have just studied that the heme consists of two parts, that is the iron containing part and the pigment containing part, known as the porphyrin. And the globin molecule is then used or utilized again for the resynthesis of the hemoglobin. So that means it is continuously reused, and the iron is stored in the body as ferritin and hemosiderin. This is important. The iron is stored in the form of ferritin and hemosiderin, and the porphyrin is converted into a green pigment called the biliverdin, which is very much important. We will be discussing more about the biliverdin in the gastrointestinal system, because this biliverdin has a very important role to play in our digestive system as well, digestion of the food, and also abnormal increase in the quantity of the biliverdin can lead to certain condition known as the erectus or the jaundice, and there are much physiological things to happen. So we'll be discussing all of these in another video. So as of now, just understanding that, summarizing it, hemoglobin gets degraded into heme and globin part, where globin is used for the resynthesis of hemoglobin, and heme part is again degraded into iron and porphyrin, where the iron is again split into ferritin. It is split into ferritin and the hemosiderin, and finally the porphyrin is converted into a green pigment called the biliverdin, which travels back into the liver, and certain chemical changes or physiological changes happen there. We'll be discussing that in another video. So with this, we come to the end of today's topic, that is the formation of the hemoglobin and also the fate of hemoglobin. If you like this video, do hit the like button once and comment down if you have any doubts. So we'll be seeing you, or I'll catch you up in another video. Thank you.